In this unit, we will discuss how DNA is able to store and transmit genetic information. This unit will be broken down into three key topics: the experiments behind the discovery of DNA as the genetic material, the mechanism of how DNA stores genetic information, and the process of transcription. This video will focus on topic 1, the experiments behind the discovery that DNA was the genetic material of the cell, and experiments that determined the structure of a molecule of DNA. Now you might be thinking to yourself, everyone knows that DNA is the genetic material of the cell, but up until the 1950s, most scientists hypothesized that protein was the genetic material, because proteins could be much more complex with 20 different building blocks, compared to just 4 nucleotide building blocks of DNA. This just goes to show that not every hypothesis is correct. One of the first experiments to show that molecules can transfer genetic information from one organism to another was completed by Frederick Griffith in the 1920s. Griffith was interested in infectious diseases, and he studied a bacteria called Streptococcus pneumoniae that causes pneumonia. Griffith noticed that the same bacterial species sometimes looked very different when it grew on a petri plate. He described one type as smooth because it formed large flat colonies, and the other as rough because it formed small raised bumpy colonies seen here. Griffith wondered what the difference between these two strains of bacteria were, so he took them and injected them into a mouse. First, Griffith injected the smooth bacteria into the mice. After a few days, the mice contracted pneumonia and died. He then injected the rough bacteria into the mice, but after injection, nothing happened, and the mouse remained healthy. The smooth bacteria are called virulent, meaning that they are able to cause disease, and the rough bacteria are non-virulent. They did not cause disease. Something had changed between the smooth and the rough bacteria that caused the pathogen to no longer cause disease. Griffith's next experiment was to kill the smooth bacteria and re-inject it into the mouse to see if it was still virulent when dead. He learned that it was not. Griffith's big surprise, however, came when he took the heat-killed smooth bacteria and mixed it with living rough bacteria. He injected that mixture into the mouse, and the mouse got pneumonia and died. On the next slide, take a minute to think about why Griffith was surprised by this result. Griffith was surprised because neither the heat-killed smooth bacteria nor the living rough bacteria killed the mice individually, but when mixed together they were lethal. He concluded that there must have been some material in the debris of the dead smooth bacteria that contained genetic information that made the smooth bacteria lethal. That genetic material must have been picked up by the rough bacteria, converting them to smooth and giving them the genetic properties that allow them to be lethal. This conclusion was supported by the fact that when the dead mice were dissecting, living smooth bacteria were found. This process of transferring genetic material is now called transformation, and while Griffith did not know that the genetic material was DNA, his experiment opened the door for other researchers to repeat his work in order to identify what molecule was being transferred. Griffith's experiments were followed up by the research group of Avery, McLeod, and McCarthy. Unfortunately, at the time, the majority of scientists believed that the genetic material was the protein of the cell's chromosomes, not the DNA. Thus, many did not agree with Avery's conclusion that DNA must be the genetic material of the cell. Scientists did not agree that DNA was a genetic material until after scientists Hershey and Chase completed their experiment using bacterial viruses called bacteriophages. Watch the video on the next slide to learn about their experiments.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
Once it was understood that the genetic material of the cell was the DNA, there was a race to determine what the structure of DNA was. Erwin Chargaff was the first to show that the percentage of DNA composed of adenine bases was equal to the percentage of thymine bases. Similarly, the percentage of guanine bases was equal to that of cytosine bases. This data was used by Watson and Crick to predict the common base pairing rules of adenine base pairing to thymine and guanine base pairing to cytosine that we recognize today. Watson and Crick also used data obtained from Rosalind Franklin, who generated X-ray crystallography pictures of DNA that identified it as a helical structure. Watson and Crick used information from these researchers and others to correctly predict and build their double helix model of DNA. A double-stranded molecule of DNA looks like a twisted-up rope ladder, seen here with the deoxyribose sugars in purple and the phosphates in orange, making up the sugar-phosphate backbone. The nitrogenous bases interact in the center of the helix to hold the two strands together. If you take that twisted molecule and unwrap it so it lays flat, you observe the figure on the right. Take note of the orientation of the sugar-phosphate backbone. One strand runs from the 5' phosphate at the top down to the 3' sugar molecule, while the other goes in the opposite direction. The 5' phosphate is at the bottom, up to the 3' sugar molecule. This is necessary for the hydrogen bond formation between the bases and gives DNA its anti-parallel structure. Often when writing DNA, we use simplified structures. You will see DNA written as just two parallel lines with the 5' and 3' ends properly labeled. These lines represent the sugar-phosphate backbone, and the bases are implied but not drawn. Alternatively, you will see the bases themselves written out, showing their complementary base pairs, and their 5' and 3' ends properly labeled. In this depiction, it is the sugar-phosphate backbone that is missing but implied. Note that in either instance, the 5' and 3' ends are labeled. The orientation of your DNA strands is very important for the proper retrieval and replication of the stored genetic information in the DNA.